Hello and good evening, viewers. Welcome to another episode of Career Mantra. I'm your host, Nltv, once again back with yet another interesting career option that is currently in demand among many. In our previous episodes, we have covered different career prospects like applied psychology, paramedical sciences, computer science, sociology, hospitality and tourism management, and microbiology. And in today's episode, we will be talking about the career prospects in the field of biotechnology, which is nowadays very much in demand as a career option. So to let us know more on this, we have in our studio Dr. Rajiv Sharma, Professor, Program of Biotechnology from Assam Downtown University. So welcome, sir.
Thank you very much.
Okay. All right, sir. Let's begin with the biotechnology today. And speaking about biotechnology as a career, well, I want to know, and also the viewers are very much keen to know about what biotechnology is. Would you please start with the introduction of the biotechnology?
Yeah, very well. Thank you very much. So let me start what exactly is biotechnology. See, it's a science which deals with the various aspects of molecular mechanism which has been going on in an organism, or which has been going on in a cell. If we talk about, then a cell can represent a life, right? So we want to understand what exactly the different mechanism goes inside the cell. Now when we know about this mechanism and understand the mechanism, harness the information, can we expedite those mechanisms, or can we carry out some manipulation in this mechanism so that the work by the organism grows very fast, or it does very fast, so that it can be benefited to the humankind. Say for example, let us take medical science, right? Let us take a medical science. We have been infected by a number of organisms, number of times. We want to know about that microorganism. How to know it? There are different mechanisms different techniques by which people started identifying the microorganisms. Like we grow them, we look into their features, then understand about that the characteristics, how it is been growing. Slowly and slowly, when we went deeper into the organism, then we started learning that it is made up of different kind of molecules, and now we wanted to know about these molecules, how it is, what are the different kind of molecules, right? Now, very simply, to identify an organism today, it is very easy. Say it is very easy to identify the organism. You take the organism, extract its 16S ribosomal RNA. Gene, that is that gene, you sequence it, you will come with some letters, information, and then you you can match with the existing organism, and you can identify it very easily. So it's very important to know the molecules which are there, exist inside the organism, and which are building the organism. And secondly, the mechanism. I will talk about the mechanism because these are very interesting prospects, like how we harness this mechanism, understand this mechanism, and use this mechanism for benefit of mankind.
Okay, obviously, sure. Sir, thank you so much for giving the brief introduction about biotechnology. Let's also know more about what is the importance of biotechnology in modern education. Sir, would you like to continue?
Yeah. Sorry. So actually, see the importance, if you look into, I just now I told you that we try to learn about the different molecules which are been participating for the life of the organism. Let us take a simple cell. The cell itself is unicellular. It is consisting of one cell. It is alive. Say bacteria, it is alive, and it is having all sorts of mechanism what we do in our day-to-day life. It eats, it excretes, it reproduces, and many other activities the single cell used to perform, right? Now biotechnology assists us and help us to understand how exactly these processes are been going on. Now if you talk about the importance, importance of biotechnology are in various fields like medical, agriculture, then your even petrochemicals, then veterinary science, plant science. So you talk about all different sciences, biological sciences, or all different important subjects. This biotechnology is somehow is been related to each other. Now, let us take, just now I have given you an example of medical science. Let us go through that example itself. Now, say I went to a doctor, I am been infected by some microorganism, and doctor simply asked me some symptoms. What are the symptoms? Whether you are having fever, whether you are having other problems or not. So, on the basis of the symptoms, in early days, doctor used to provide you a medicine. Fine. Now, when we came to know about the molecules, later on, and the microbiological techniques developed. After that, microbiological techniques developed. Now, if you go to a doctor, you visit and you have been infected by some organism, so doctor will ask you to go for certain kind of test. Either it will be a biochemical test or it will be the culture of the microorganism. Culture will provide again the identification of the microorganism. That also is a primary understanding about the microorganism. When we didn't know about many different type of microorganisms, there are millions of microorganisms within our gut itself. There are it is more than our cell. Now today, if you visit a doctor will prescribe, sequence the gene of this microorganism, identify the microorganism, so you will get a correct identification now, because we are known to many microorganisms, genes of these microorganisms are already known. So you know that there are many different databases which are being built. Say for example, NCBI, National Center for Biotechnology Information. So where already known gene sequences, DNA sequences or protein sequences are already been there. Now, if I find a sequence, I will match my sequence with the database sequence. There are some methods by which we can match, and matching if it provides us the information that this sequence matches with some XYZ, so we can easily identify that organism. Now, this is in the case of identification is the primary task, right? When you study microbiology, when you study say virus. Now take the example of COVID-19, right? When you suffer from some fever, so usually swab is being taken or nasal fluid is being taken, and it is been exercise. The same exercise whatever I told just now, you go for RT-PCR, right? That's a different mechanism, but the same technology goes for identifying that COVID-19. So this is the primary task of a biotechnologist or a microbiologist or a biologist. So to identify the organism, right? Next, number of ways in medical science. Say we are acquainted with our food, so we take our food, we digest it, get energy, and we can do work, right?
Yes.
It's a very simple concept. Now say during this process there are a number of molecules which are being released, right? Released, and these molecules perform different functions. Fine. Now for example, we do lack vitamin A in our body. What if a plant, if it is been grown in a way so that it can provide me vitamin A, right? So you use biotechnology, manipulates its genome or genes, then grow the plant, the organism will develop some vitamin A, you eat it, the vitamin A will be in your body. The best example is the golden rice, right?
Yes.
So golden rice is providing you vitamin A. Similarly, now this is a technology which has been applied in the field of agriculture. Similarly, in veterinary science, say white revolution, you need huge quantity of milk. So there are some experiments which were being carried out to enhance the milk from the animals, so that the mankind can be fed with this milk. So other experiments like in the field of pharmaceutical science, in the field of medical science, I told already, agricultural science, veterinary science, in marine life. So number of ways or number of means are there, a number of important applications are there in various fields of importance. These are also very important fields, like medical is very important to us.
Of course, yes.
Agriculture is very important to us. So similarly, veterinary is very important to us. So all these important fields can be regulated or can be enhanced, can be studied in more perfect way when we understand the molecules of the organisms. So that's how it is important. Biotechnology is very important in every biological science you can talk about, or in another way you can say that it is a tool to study all the biological fields. Say I am a botanist, I want to know about the plant, biotechnology is my tool, I apply biotechnology and I can understand the plant very well. I am a zoologist, I want to know about the animal, so similarly I apply biotechnology and I can understand very well about the animal. So similarly, the human again is the case study which Human Genome Project started in 2002. I mean, it was been completed in 2002, and the entire genome sequences is known now. So we understand various problems of human being, right? Some genes which are responsible for causing diseases, some genes which are responsible for other activities which are important requirement of the human being. Say for example insulin, right? Insulin, if it is not produced in the body, we need to inject insulin in our body. So now insulin, where from you get the insulin, right? Now insulin is been produced by some microorganism. How? I take the gene of from human body which is producing insulin. Now I fix it in a microorganism. Now that microorganism, when it's been grown, it is producing insulin. I am extracting that insulin, I am using this insulin, right? It's a very simple example. So there were other techniques also for the production of insulin. Earlier it was being used different ways.
Okay, viewers, by now we have understood what biotechnology is, and to make it short and to tell you what biotechnology is, it is the study of biology and technology, as we have all understood. And Dr. Rajiv, he has already elaborated how biotechnology works in different fields like medical, either it be veterinary or either it be in just to discover a virus, or different fields that it comes up to, and also with the help of microorganisms, different medicines or different different tablets or different things that have come up has helped people even in agricultural field. So let's also know about what are the core contents to become a biotechnology, sir.
Okay, what do you need to know to become a biotechnologist? Now, to become a biotechnologist, we need to know about the biological science which are already been existing. Together with those information or those knowledge, right? Say, I am a microbiologist, I am a botanist, I am a zoologist, or I am a person from medical science, or I am a person from veterinary science. So I am in a field of biology. I am already trying to understand some of the aspects of biology. Now, to this particular biological content, if I add this technology, right, add the technology, simple technology of how we would be able to understand the mechanism which has been going on inside the cell. Now, at this moment of time, one of my cell is working it has been working in a very different, a number of multifaceted, work. It is producing enzyme, it is replicating, it is producing RNAs, and it is producing different kind of molecules, right? A big biochemistry is going on inside my cell. Now, to understand this, we need to know, these molecules, we need to know basically the DNA, which we call deoxyribonucleic acid, the RNA, and the protein. These are the core content in molecular biology. This core content, to understand this core content, we have different processes, right? Different mechanism by which we try to extract this DNA, understand this DNA by sequencing it, knowing in what form exactly it is. We extract the protein, again we sequence it and understand how the protein is, or what will be the structure of this protein. So many ways we go for it, and if you look around, basically 2, 3, 4 instruments are there which, or techniques are there which enables these processes, like DNA sequencing. Say if you go for whole genome, so there are a number of techniques like described and mechanisms are being known. Say for example, whole genome shotgun sequencing. It is like gun, you break the DNA, fragment it, then you carry out sequencing of this DNA, and all these fragments you again rearrange this DNA and get the whole genome. Let me tell you about our genome. It is 3 into 10 to the power 9 nucleotides. That means if you write down in the form of letter A, T, G, C. So if you arrange it in the form of letter, you will require for a single genome, for my genome, it will require 300 Oxford Dictionary, right? Of that format and of that size, and of that letter size also the print, if it has been printed. So it is a very, very large molecule, right? Now, we take this molecule, understand this molecule, now we carry out extraction, then we understand, then we think about how to manipulate these molecules. We can carry out genetic engineering, what it is called. Genetic engineering, manipulation, then we use this engineered gene into some organism, say for example, to create a transgenic organism. Like for example, earlier I already gave an example to you earlier, like BT brinjal, right? Then golden rice, then BT cotton, right? So BT cotton and BT brinjal is a gene which has been extracted from an organism called as Bacillus thuringiensis, and it has been introduced to cotton to produce certain toxic substances. How it has been done? We studied the molecule of Bacillus thuringiensis, then we have gone across understanding it, how it function, then we applied the gene to some other organism like cotton and brinjal, and the plant is now performing the same function. Say it is producing a toxin which is responsible for killing of some insects. Now when the plant has been grown, insects cannot attack that plant. So it's an application again in the field of agriculture. So the process is very simple. Understand the molecules, understand the function of these molecules, then apply the function of these molecules, or apply these molecules and look into whether it functions similarly in all different organisms, or it works differently in different organisms.
Sir, would you like to say about the courses provided in your university?
Yeah, in detail, right. Say Assam Downtown University was established in 2010, and the biotechnology started from 2012 and 13 UG and PG programs. Now, today it is about 10 to 13 years, which is 12 years of the growth. Now, in biotechnology, we are providing two different programs like BSc Biotechnology and MSc Biotechnology at present.
Okay.
And all the allied fields, like just now I told you about that there are some allied fields to be known, including BSc Biotechnology and MSc Biotechnology. We also have the programs of microbiology, BSc and MSc, then food technology. I forgot to tell you about, very interesting, that biotechnology is very, very useful in food technology. Right. Food technology, that is called as FND, food nutrition and dietetics. Then we have zoology and botany, which are the conventional subjects whose knowledge is very, very basic for being a biotechnologist. Then your microbiology, I already told you about microbiology. We have biochemistry too. So these are various programs which are also there with biotechnology. Not only biotechnology, but these are all programs which are there in, all fields are there. It doesn't come under, it's providing some different degree. But to become a biotechnologist, these fields are also to be known.
Okay.
Some of these fields, say if I am a botanist, as I told you, that I am a plant scientist, so I want to use biotechnology as a tool to improve my science in plants, or manipulation, or working with molecules in plant. So we have the program that a student can go through plant science as well as your biotechnology. Even they can go for zoology and biotechnology. Even they can go for
even after taking these courses like zoology, biology, they can still study botany. Botany.
So
they can
if you talk about, you were asking about what called eligibility criteria, how to become a biotechnologist.
Okay.
Now, in different levels, there are different levels, like graduate level and postgraduate level and PhD levels, right? Now, to be biotechnologist graduate, so you require higher secondary in science. PCB, if it is BTech biotechnology, then you will require also mathematics together with PCB. Now, BSc biotechnology, in our university, we are offering BSc biotechnology where it requires PCB as a major basic component, which will be very, very important requirement in the field of graduation. Now, science graduate, basically we say that science graduate can go for science, higher secondary science can go for this BSc biotechnology. Now, to become a biotechnologist, MSc biotechnology, the program MSc biotechnology, the student, if he comes from BSc botany, then also he can go. If students come from BSc zoology, he also can get admission in biotechnology. Students from BSc microbiology can also join MSc biotechnology. So there are number of entry points you can talk about. That MSc program, you can handle many different subjects, right? Graduate courses, right? Then for the PhD program, we also have a PhD program. Now, for the PhD program, it's not mandatory to become a biotechnology, MSc in biotechnology. Now, as I said, that this is a tool which enables us to understand the molecules. So, if I am a MSc zoology, if I am MSc botany, MSc microbiology, even then I can use biotechnology as a tool and carry out my research work. And very well I can have some findings on the molecules and understand about these molecules, and so that we can apply these molecules for various benefit of mankind.
Okay, sir. So, talking about, I believe we have almost covered this eligibility criteria.
Right.
Still, if a student has completed his higher secondary studies in BSc, so like you have mentioned, it could be either botany or zoology or different subjects.
Field,
So if he has finished his higher secondary studies and if he wants to join Assam Downtown University to continue his studies in biotechnology, then what are the eligibility criteria for that student?
That eligibility criteria is if he has done in any field of biology, it may be zoology. You are talking about graduation or you are talking about post-graduation?
About both UG and PG.
UG and PG, right?
You
should be higher secondary science, right?
Yes.
Higher secondary science, biology as a component.
Yes.
In that science, then you can join biotechnology. If you are I mean for BSc biotechnology, right? If you are BSc in biotechnology, if you are BSc in microbiology, if you are BSc in botany, if you are BSc in zoology, so all these are biological field, so you can join post-graduation in biotechnology, right?
And what is the marks that a student should have?
Say it's the eligibility criteria marks is been recorded, it is 45 to 50% that is required for, that is minimum. If you have better the marks than that, it is very easy to opt the course, yes.
Okay, all right, sir. Okay, and also, sir, what about PG? Those students who would like to continue biotechnology in Assam
PhD or PG?
I mean,
PG is postgraduate.
Yeah, postgraduate
Post-graduation,
courses.
just now I told you MSc in biotechnology.
Okay.
So this is a 2-year course. So 2 years program, 2 years program. So it includes 4 semesters. So in every semesters, the students has to appear for examination and carry out good mark to pass the examination. He has to pass the examination. Finally, he will be out with the degree of post-graduation in MSc, called MSc in biotechnology. After 2 years, right? After 2 years, he will be, if he secure the eligible marks or eligible, grade, so he will be awarded with the degree MSc.
Okay. So viewers, we have heard that it is easy and it is also applicable for those students who have secured merit marks, and it would be very easy for them to get through Assam Downtown University to study biotechnology. However, those students who require at least 45, right, sir? 45
to 45 to 50%.
Yes, 50% also, they would be eligible for this course. So moving further, let's also know more about the teaching methodology of Assam Downtown University.
Yeah, before that, let me tell you more about biotechnology, because your career, your career development, say a student should join any of these courses, right? Any of this program or any of these courses. So whatever the interest is there, whether he is interested in understanding the molecules or not. So interest is the most important criteria. Besides these academic criteria, right? I am telling you just for, it's very important. Say interest in the field is the most important criteria. First, you should have the interest. Then you should have your eligibility criteria. I forgot to tell you exactly about the interest. So it is very important to have interest on which field the students want to go or want to move. In some cases, you will find that once a higher secondary student learned his biology from me, so he is now in US. So he has 2 problems. He didn't have any interest in biology. And secondly, he had a problem in writing English, and again a problem in drawing. So he came to me and asked me what I can do about this. So I started telling him stories of all those molecules, how it functions, how it works, and it is very interesting to know all these phenomena which are going on inside the cell while I am sitting. Now, when these stories were being told to him, he became interested in biology, okay, and he scored a good mark Now he did his medical. Now he is doing research in some genome and other activities like genomics and all. So he is doing, now he is in US. One of my student, he learned bioinformatics from me. So that student also was keen to understand how is the molecule functions. So biotechnology and bioinformatics, both these two goes side by side. They are interrelated to each other, because if you understand biotechnology, you perform experiments in laboratory and extract the molecule, look into the molecule, how it functions. Now bioinformatics is a way where you take this molecule to a computer system, right? Where you perform analysis, where you perform all other, analysis like applying some mathematics, right? Where you understand all these molecules and then bring it out again, the information, use the information for the benefit of the human being. So this is, what I want to say, that all the biological fields, one way or other, are interrelated to each other. So it is very interesting, but you should have the knowledge, have the interest. You should have the interest to work with molecules, understand the molecules, and apply these molecules for the benefit of mankind.
Okay. So before discussing about the facilities provided by Assam Downtown University, let's talk about the teaching methodology provided in Assam Downtown University. So, sir.
Okay. So teaching methods, say, if we look into the history of teaching method, so there was a Gurukul system of teaching and learning method, right? So exactly, it is not only the teaching method, it is teaching and learning method, right? Now, slowly and slowly the things has been changed, and now we were in the classroom, and we are in a room having 4 walls, and a teacher and a student interact with each other. So looking into this perspective, right, how to go for teaching them in different ways so that they can have an experiential learning. So what we did, we have improvised our courses, and we tried to improvise in a way that more of experiential learning than the theoretical knowledge gaining starts. So more of experiential learning and activities based teaching or activity based learning starts on. So what we are doing right now, that activities such as they are asked to go for writing assignments, they are asked to present what they have learned, or we simply give a topic to them and the student prepare themselves, prepare and bring it to us, try to deliver it. So we find some problems, so we discuss with them. So it is interaction based learning, right? Interaction, we discuss, and then finally we come to the conclusion what about. So we have a defined policy right now in Assam Downtown University, how we are going to perform the teaching and learning method, and also it includes the assessment method, right? How we will go for assessing our students, and how what will be the method of assessment, and how we will find out which of the student is poor in learning, which of the student is good, and what are the different, different way to force them. One has to move from poor learning to a good learner. Another is already a good learner, so we want to enhance them to a good, say, biotechnologist, microbiologist, or FND. So whatever it may be the program. So we have a defined and focused way of teaching and learning method. So teaching as well as learning both go simultaneously, because even if a teacher has to learn the things, then only he can deliver to the students. Yes. So we have a definite structure, and we focus on experiential learning in Assam Downtown University.
Okay. Sir, any addition to that question?
So that is what the ways we go for practical based learning. Experiential learning, I mean to say, we increase the practical content of our syllabus. We increase the activity learning, like their presentation, like their questioning. Even say sometime you will see that the students come to the class, they sit, the teachers goes and talk, students simply nod their head, and the teacher come out, and there is no interaction takes place in between the student and teacher. To break those, we have very young faculties who are friendly, they are mentoring the students, they are working together with the students. Say if the student is very good, they used to provide some research work, they carry out their research work. Those who are a little bit poor, they are being taught in tutorial classes. We have some tutorial extra learning scheme. And also, if the student need based learning, right? If the student asks me, I do not understand this thing, so anytime they can come to us, come to the teacher, and we are we discuss about the things, and we try to solve his or her problem. So anyway, we focus that the students should learn something. Yeah, that's how in Assam Downtown University we carry out the work, the teaching and learning process.
Okay, all right, viewers, we have heard from Rajiv sir about the different teaching methodology of Assam Downtown University. However, he has also said that it is not about one go, but it is about two go, where both teaching and learning simultaneously go hand in hand. So now we will talk about the different facilities provided in the university.
Okay. Yeah, different facilities of say teaching and learning. You talk about, say, yes, obviously there are classrooms, there is library, there is laboratory, there are laboratories, approximately 12 laboratories are there for our program, because it is an integrated program, right? It is having MSc Biotechnology, Microbiology, Biochemistry, Zoology, Botany, so all the labs are there, so it includes entire lab. All students can access any of this laboratory, right? It is not demarcated that this is the lab for biotechnologists only. So this is free accessible, and all students can learn in any of this program. They can access this and use this laboratory to work out. Now this is all about teaching and learning facilities. So ICT enabled classrooms are there, then seminar halls are there if we have to interact with, say, students of all the programs. Some courses are there which includes all the students of all the programs, right? So in that case we require bigger sized room, and we require facility to talk, right? Loudspeaker and mic. So in that case, those kind of rooms are also there. As a biotechnologist, I told you just a little earlier that bioinformatics and biotechnology goes hand in hand, right? Now, bioinformatics is a computer-based, so we have a computer laboratory too. Library includes departmental library in the we call it Faculty of Science. So inside the Faculty of Science library is also there, and the central library is there. Further, these are all relevant to your teaching and learning requirement. Besides these, the student has to grow itself mentally, right? These are the facilities for mental growth. And the students also have to grow itself physically. So we have facilities like swimming pool, international swimming pool, international football field, volleyball field, your basketball court, badminton, all the sports facilities you talk about. So all the sports facilities are available in our university. So after say 3:30 to 4:30, the students can freely go for your sports and other activities. Some of the students like to have music, even music is there. Some of the students may like to have your co-curricular activities like your extempore speech learning or debate or many other things they can go for. So to create an environment like this environment, we have clubs. We have introduced a club system. There are many clubs like your sports club, your music club, then art and craft club. So in that way we have different groups. All those groups participates in some kind of activities very frequently and carry out their learning not only in, say, in one field but in all aspects. Both, if we concise it, it is both mentally and physically, right?
Yes. Very well. Now, viewers, we will be talking about once a student or once he finishes a degree in biotechnology, what are the different prospects of acquiring job?
Yeah. Yes, it is very important question to be known by the viewers. And Emily, you know, this is why a student choose a program. So he starts a career in a particular field. Say after some period of time, I must have a job in my hand after doing this. So to get a job, there are different stages of learning. Like we have divided the programs under different levels, say higher secondary, then graduate level, then postgraduate level, then PhD level. Obviously, it is true that when you are in a PhD levels, that is the higher end of your education system, right? Further you can go for, but formal degree ends there in PhD. Now, if you are a PhD, then your prospect is much higher than a graduate. If you are a postgraduate, this also is higher than a graduate. So, at different level, there are different prospects. Now, all around, if we look into in the field of biotechnology, if you go for finding out prospects, approximately there are more than 300 companies, right, which are working in one way or other in the field of biotechnology. Now, what are these companies? Say, pharmaceutical companies, a very best example, Ranbaxy, Cipla, then your Drs. Reddy, right? And you can look into different companies who are working on this. Then agricultural companies are there who are say producing what called biofertilizer. A biofertilizer producing company may require a biotechnologist because they want miracle, right? Say I am using a microorganism to enhance the fertility of soil, and I want that it has to be done in a very good manner, and it is to be speedy enough so that the plant get nutrients and they can grow very well, right? So if you talk about agricultural farms or agricultural industries, right, who are producing biofertilizers, who are producing, just a little while ago I gave you an example of BT cotton and BT brinjal, right? So to preserve, understand about BT, now you need a biotechnologist. Then there are a number of other avenues where this biotechnologist may go for job prospects. Besides these companies, right, there is a job, what I am doing, academics. You can become a teacher. You can from the school to university level, you can be a teacher in various level, various stages, but for being a teacher, your requirement of knowledge or your requirement of degree or eligibility criteria may differ, right? Now coming down, say I'm a graduate in biotechnology and I want a job, what I can do? So there are a number of laboratories all around India. If you look into these laboratories, most say pathological laboratories, a very simple example, and it is very widely available laboratory. Now in pathological laboratory, say during the COVID period, what was the most viewed test which was been done. The laboratories are to go for COVID test. A biotechnologist is required for performing this COVID test, right? The biotechnologist has to know about extracting the RNA and then go for polymerase chain reaction and then go for it is called as RT-PCR, or everyone knows now RT-PCR, what is RT-PCR is.
Yes.
Right, reverse transcript is PCR, polymerase chain reaction, right? It has been known now to all, and that is given by COVID, right? The term is been known.
Yes.
Earlier, only the academicians know about RT-PCR, what is an RT-PCR, right?
Okay.
Now every individual from a very, it is common word now. So these are to be performed, and this can be done by a technician. We call it as a laboratory technician, right? So you can find your job in various aspects according to your, according to your level. How much knowledge you have acquired, what are the degrees you have acquired. So most important is the knowledge, and secondly, everyone when you go for a job, it will naturally go for looking into what you have done. So both these two combinedly, you can have a better opportunity.
Okay, very well, viewers. That will be all for today. However, before we wrap up, thank you so much, Rajit sir, for joining us.
Thank you.
And also, to know more about such career prospects, keep watching Career Mantra only on Nagaland TV.